after finishing his first MD degree, he did a PhD in which he discovered the peptide endothelin-1, which was quite impressive. But then he went on to subsequently discover orexin, about which he's going to be speaking today. No coincidence, I think, that he's done that, and to have discovered two druggable targets in different areas of science is extremely impressive. I've known Masashi for over 30 years. It's always been a pleasure. He's a great colleague and a brilliant man. So over to Masashi.
Okay, so what should I do? I need some help, I guess. Can somebody help me? Simply.
Come over, shouldn't they?
I don't think I see it.
Don't, no. Just close that. They're going to put them up.
Oh, okay.
Do you have the slides at the back? Wait a moment.
Okay. It says on the screen, wait a moment. No, usually, I almost never let my slides copy to another machine because it leads to lots of troubles. And this time I had to. And quite expectedly, there are lots of trouble in many slides, unfortunately, which took me a lot of time. And it wakes me up completely from this jet lag, which was good. I'm actually going to talk about that in the talk. No. Should I have
If you have your laptop here, we can maybe patch you in.
Oh, that's. Oh, that's the easiest way, actually. Okay. Let's see. Nope. Recognized. Oh, here it is.
Do you want me to just withdraw this for a moment?
No.
Bring up your slides.
Yeah, my main screen was here.
So can you bring up your slides?
Yeah, okay. Let's see. Yeah, this is the slide. Okay.
Then if you go to presenter view, so if
Yep.
you go to here, and you try this.
Yeah, no. This is my problem. Yeah. There's no signal. Almost there. Almost there. Good. I think we solved the problem almost. Great. Finally, thank you for waiting. I'll be happy to present my talk from my own computer in the end. Okay, so as David introduced me, so I'm going to talk about the neurobiology of sleep in the next 45 minutes or so. So this is a quite busy slide, but tells you two simple things. One is, by the way, this is all terrestrial mammals in the order of phylogeny. That's important, in the order of phylogeny genetic tree. And the vertical axis is their average time of sleep per day. So even from the distance, you can see two things. One is, although the species difference of sleep time is great, from 3 to 4 hours to nearly 20 hours per day. However, there is no species which does not sleep. That's one thing. The other is the sleep time tends to be closer when the phylogeny is closer to each other. So they call it a phylogenetic signal in sleep time, which suggests that the daily set point of how many hours you sleep for that species is set by something in their DNA. That's the suggestion. And that presents two major big questions in the field. Number one, why everybody sleeps, even though if you think the sleep is quite risky behavior for wild animals. Why do they sleep? The other problem is how, for example, the set point for daily sleep time is determined. Those are two big questions in the field. That was a mammalian species, but now the consensus in the field is that every vertebrate sleeps, down to fish and chordates. And even invertebrates, like insects or worms or octopus, whatever, they also sleep. The problem is, their brain and our brain is completely non-homologous. The brain is clearly invented more than once. So where's the origin of sleep then? So several years ago, these three students in Caltech published an amazing paper which tells you that jellyfish sleeps. That's amazing because jellyfish doesn't even have a brain. They have only a kind of distributed nervous system. There is no CNS there. However, they already sleep. So the conclusion of this paper is that sleep is invented before the brain. Okay? So sleep is such a fundamental behavior. So like I said already, there are two big questions in the field, and it remains a great mystery. So with regard to the regulation of sleep, one subfield has seen a great progress in the last 10 years or so, thanks to newer technologies like optogenetics and pharmacogenetics. So the conclusion, the current consensus is that sleep-wake is switched in the entire brain through this neural circuitry-based mechanisms. So the consensus is that there are different classes of neurons promoting wakefulness, and they have this wake-promoting neuronal circuits or networks. On the other hand, there are sleep-promoting neuronal networks in different locations of the brain, and overall, they compete with each other. So they are either indirectly or directly inhibiting each other, creating this flip-flop-like meta-stable switching circuitry. Okay? So this much is known. In other words, this sleep-wake state switching is executed by the neural circuits. And one of the representatives of this wake-promoting system is the orexin-producing neurons, which we discovered more than 20 years ago. So the orexin neuropeptide was initially discovered as a ligand for orphan G protein coupled receptor. So initially we did not know the physiological function of orexin at all. So initially we even hypothesized that, or published a paper with the claim that orexin main action is to control appetite. Actually, Greek word orexis means appetite. However, when we generated then the orexin knockout mice, we did not see any decrease in body weight or appetite. Rather, orexin deficient mice are apparently completely healthy, happy, and behaving normally, apparently. Until it occurred to me that mice are nocturnal species, so we should observe the mouse behavior during the night, in the darkness. So we set up infrared video here. And by the way, this is EEG and neck EMG of this mouse, which is clearly awake right now. And he's grooming his face right now, clearly awake. However, in a few seconds, this mouse completely stopped moving, and actually he fell down to the floor. And simultaneously, this EEG pattern changes to this theta rhythm dominant REM sleep pattern, and neck EMG disappears. So what happened then was direct transition from wakefulness to REM sleep-like state, which should not happen, never, in healthy animals or humans. And within a few seconds, he wakes up and behaves as if nothing had happened. So this was very weird transition from wakefulness to REM sleep, which already suggests that they suffer from the disease called narcolepsy. And then Emmanuel Mignot in Stanford, they were keeping this autosomal recessive heritable form of dog narcolepsy. So this is Emmanuel Mignot, and this Doberman, they go through the same type of attack as you saw in the mouse. Here is our mice. These dogs, see, when they are playing together, they lose muscle tone and go like this. So this is dog version of what's called cataplectic attacks. And he positionally cloned the mutated gene, and turns out that it's one of the orexin receptors. So that was a very dramatic moment, scientific moment, in that we went from the gene and discovered that knockout mice have a narcolepsy. They went from the disease in dogs, and turns out the mutated gene was the receptor for the ligand. And then, even after that, Emmanuel went on to show, importantly, that human narcolepsy patients have lost their orexin-producing neurons in the lateral hypothalamus here. It's a very specific loss of neurons. Other kinds of neurons in the same area of the brain is completely normal. So human narcolepsy is now defined as an orexin deficiency syndrome. This is an example of a patient with orexin deficient type of narcolepsy. This woman, she is actually, you see, smiling, and that triggers this cataplectic attack where she bilaterally loses muscle tone. This is a intrusion of REM atonia mechanism into wakefulness. That's the interpretation. So again, she's, so her boyfriend is smiling right now. She's laughing, and then that will trigger this attack. So it's a very serious disease for the patient, but this is caused by the loss of orexin. So that, to make a long story short, makes the orexin great target for drug treatment. Now the orexin antagonist, if you block orexin, you will be sleepier because orexin is essential for keeping you awake and maintain your wakefulness. So orexin blockers have been already approved as a treatment for insomnia. There are multiple compounds available now clinically. Orexin agonists, on the other hand, should be a mechanistic treatment for narcolepsy and perhaps other types of hypersomnia conditions. And we also contributed in publishing the very first small molecule orexin receptor agonists back in 2015. But right now, the forefront is by Takeda company. This compound is now phase 2B clinical trial. So we hope that this will make a drug in the very near future. So the orexin system, bilaterally, I would say, the positively and negatively could be a drug target. So as I told you, this trouble of my PowerPoint slide completely wakes me up from this jet lag. So, another lab in the same institute as I am, Mike Lazarus' lab, has been studying this very interesting phenomenon of, sleep gating by motivation. So, when you're highly excited, you forget about sleepiness, right? So his story started with adenosine, which is known to be a very potent somnogen in the brain. And caffeine is known to be an antagonist for the adenosine receptor. So that was known classically for a long time. What they discovered in the last 10 years or so was where in the brain this adenosine and the caffeine works to regulate sleep. Very unexpectedly, the target was nucleus accumbens, this indirect pathway cells, part of which express A2A receptor. So these neurons, nucleus accumbens neurons, are very potently inducing sleep-promoting neurons, and adenosine stimulate that neuron, caffeine blocks it, and when you're highly motivated, this VTA, dopaminergic projection to the same cell through the D2 receptor inactivate this neuron, which brings you wakefulness. So that's the mechanism for, when you are bored, you are sleepy, and when you're excited, you forget about sleep. So these makes a very nice example of this wake-promoting neuron, such as orexin neuron, and sleep-promoting neuron, such as this nucleus accumbens D2 receptor expressing neurons. So that's fine. So they make this neural circuitry-based sleep-wake switching mechanism. But what's the finger which pushes that switch? That's the deeper problem. And in the field, many people agree that the sleep pressure is regulated through two independent processes. One is so-called homeostatic process versus circadian process. So especially the homeostatic process of sleep regulation remains complete mystery. So during the wakefulness, gradually this sleep propensity or sleep pressure accumulates. And in order to resolve that sleepiness, you have to sleep. And during sleep, that's gradually released. And this is a very slow process, talking about hours to 10 hours. On the other hand, this switching machinery works in 1 second to a few seconds, both in mice and humans. So what's the relationship between these two completely different mechanisms? It's nicely depicted by this Japanese thing called shishiodoshi, this bamboo tube sitting upwards, and water is trickling in very slowly. and gradually the water levels goes up, and at certain point, it flips down. So that's the neural circuitry-based switching machinery, and once it's facing down, the water starts to flow away, and this water takes 7 or 8 hours in humans to empty, and then when it's nearly empty, then it flips back to the upward position, which is wakefulness. So, and in this particular cartoon, the orexin is depicted as a nightingale making some weight here to stabilize wakefulness. So this is a nice cartoon, and conceptually, this nicely depicts the process of sleep homeostasis quite accurately. However, what is the actual physical entity for water? Nobody knows. What is the bamboo tube, which is a counter or integrator? Nobody knows. And how the information that water filling up is transmitted eventually to the switching machinery, that's nobody knows. So this is such a deep mystery which has not been solved today, even today. So about 10 years ago, we stopped making any concrete hypothesis about this, and we started non-hypothesis-based data-driven research. In our case, in the form of so-called forward genetics conducted in mice. So what we did was to randomly mutagenize this male mouse with ethyl nitrosourea, the mutagen, and it was mated with a counter strain female, and we generated lots of these F1 heterozygous random mutants, and we screened not hundreds, thousands of this F1 mouse in terms of sleep and wake characteristic through EEG, EMG measurement. That's said easy, but it's really labor-intensive, large-scale project. And once we find a mouse with weird sleep-wake pattern, this is so dominant screen, heterozygous screen, we backcross to the counter strain, and in the next backcross generation, we could map the gene by using classical genetic mapping technique. So this is an example of a sleep recording room in our building. So these are individual mouse cage, and here mouse is nicely sleeping in their nest they make themselves. And here is a data acquisition station. So in this one room, we can record 128 mice, the sleep-wake together. So I'm going to talk about this one mutant pedigree we named Sleepy back in 2016, which is a very interesting mutant. So Sleepy mutants are hypersomniac. So they are, sleeping a lot more than wild-type mice. It's a semi-dominant mutation in the serine threonine kinase called SIK3. So heterozygous mice wake time is decreased, homozygous mutants further decreased. And importantly, despite the fact that they sleep this much, they are still sleepier. So the best marker for the level of sleep pressure at that time is slow wave delta wave density in the EEG during non-REM sleep. Unfortunately, you cannot measure delta power during wakefulness because there is no delta. But once they get into non-REM sleep, you can measure the delta density, and that's best index for the ongoing level of sleep pressure. So that sleep pressure is greater in the mutants, and they also have exaggerated response to sleep deprivation, after sleep deprivation. So despite the fact that they are such a long sleeper, they are still sleepier. It's very similar phenotype to human idiopathic hypersomnia patients. So it turned out that it was a mutation, splicing mutation in SIK3 gene, which removes exon 13, which happened to be an in-frame exon. So it skips about 50 amino acids in this protein, which includes this important serine 551 residue, which is known to be a target for protein kinase A. And this is actually, this phosphorylation reduces kinase activity, it was known. So that makes this deletion of exon 13 gain of function mutation, okay? So as you know, formally speaking, gain of function mutation could do nothing, to do with the endogenous function of the protein. So we made then SIK3 loss of function mutant, in this case in the excitatory glutamatergic neurons, because the systemic knockout kills the mice. And lo and behold, it does reduce the non-REM sleep time, and more importantly, it greatly reduces the index for the sleep pressure, okay? So it's a mirror image phenotype, more waking phenotype, mirror image from the original sleepy mutation, showing that endogenous SIK3 is indeed important for the physiological regulation of sleep. So in a sense, we made a new and probably one of the first model of sleepiness, genetic model of sleepiness. Before that, the only reliable way to make a sleepy mouse is sleep deprivation, obviously. But sleep deprivation has two problems, okay, as an experimental system. One is sleep deprivation by itself is a great stress to the animal. By the way, it is great stress to the graduate student who is doing the experiment, but stress to the mice as well. And also, sleep deprivation, by definition, the mice were awake, right, in the last few hours at least. So you cannot distinguish, oops, something ongoing. What happened? Oh, good.
Yeah, it's back.
Great. But very interesting is happening. Oops. What should I do?
It's just going to get you in the right place, I think.
Okay. It says use the clicker.
Use the right click to go forward.
Yeah, okay, thank you. All right, so anyway, what I was going to say was, sleep deprived mice were by definition awake, right? So you cannot easily distinguish the brain changes induced by wake state versus sleep state, and changes effect of increased sleep need. That's another problem. So in collaboration with Chin-Hua Liu's group in the same institute, we decided to cross-compare sleep-deprived mice. So sleep-deprived mice were sleepy because they did not sleep. Sleepy mutants were sleepier despite the fact that they sleep more, right? So we cross-compare these conditions and make a common denominator that should reflect the changes accompanied by increased sleep need. That's the logic. So to make a long story short, and what to compare, the answer was easy because the sleepy mutation was in serine threonine kinase. So we decided to do phosphoproteomic analysis in the whole brain, very simple-minded experiment. And turns out that after sleep deprivation for 1 hour, 3 hours, 6 hours, mice are getting sleepier and sleepier, and part of those brain proteins are more and more hyperphosphorylated. Okay? So the lower half is the example of such proteins with increased level of phosphorylation when mice are getting sleepier and sleepier. Turned out that many of these, oh, by the way, this is not just correlation. When you inhibit SIK centrally by this known SIK inhibitor pharmacologically, it will expectedly reduce the level of phosphorylation of certain proteins, and then importantly, level of sleep need index after sleep deprivation in wild-type mice. So turned out that many of these proteins are known to, actually 90%, almost all of these proteins are known to be localized in synapses, both presynaptic and postsynaptic. So at that time, we hypothesized that maybe the level of phosphorylation, cumulative phosphorylation of certain select proteins localized in the synapse is the one underpinning or substrate for the sleep homeostasis. And that nicely matches with this so-called synaptic homeostasis theory of sleep function. However, there are lots of remaining questions still remaining. One question is where in the brain this sleepy mutation works. So in order to answer that question, unfortunately, the SIK3 kinase by itself, this lower half is in situ hybridization histology. It's expressed everywhere in the brain, pretty much. All neurons more or less express this enzyme, so it doesn't make any answers. So we made these mice upper right half. So we fluxed the exon 13, which is skipped in the original sleepy mutation, so that sleepy allele can be recreated in a Cre-dependent manner. And we also made this mature neuron-specific inducible Cre driver. And Kanako Iwasaki mated these two mice and injected tamoxifen. And lo and behold, the original sleepy mutant phenotype is recreated. Do I have a pointer here? Laser or whatever. If not, that's fine. So by using the same Cre-dependent version of sleepy mutant mice, Hotta-san here mated that flux mice with a whole lot of Cre drivers, the brain region-specific Cre drivers. The middle panel, when the sleepy mutation allele is recreated in the whole brain glutamatergic excitatory neurons, it nicely reproduces sleepy mutant phenotype, both in terms of sleep time and sleep depth, or the index of sleep need, the bottom one. However, when you induced sleepy allele only in the forebrain, the cortex and the hippocampus, interestingly, sleep time did not change, but sleep depth, or the index of sleep need, does increase. On the other hand, the right-hand side panel, when you induce sleepy mutation in the hypothalamus, now you increase sleep time or sleep amount, but does not change sleep depth. Very interesting. So especially in terms of hypothalamic, oh, by the way, so in addition, when you mutate same SIK3 in suprachiasmatic nucleus, which is a master clock site, very interestingly, it now does change the free-running period, circadian period. Now they have a longer period and delayed wakefulness, so to speak. So the conclusion right now is the same molecular pathway apparently regulate amount, depth, and timing of sleep in 3 different brain locations. So especially within the hypothalamus, now Kanako is delving into more detailed mapping of where it works. And currently, she thinks that some anterior medial part of the hypothalamus, including, for example, ventral medial hypothalamic nucleus, BMH, seems to be a focus for the sleep time regulation. And we hope that by narrowing down such responsive neurons, we will eventually be able to connect this to the neural circuitry which switches wakefulness and sleep. That's what we are doing right now. This is unpublished results. So another obvious question is the molecular pathway. So as I said, SIK3 is a serine threonine kinase. So it does have an upstream activating kinase, downstream target. What are they? So in terms of upstream, the literature suggests that, by the way, the SIK3 belongs to the AMP kinase family. So all AMP kinase family members are under this master regulator kinase called LKB1. And SIK3 is also known to be regulated by LKB1. So LKB1 phosphorylates this T221 residue within the kinase domain. And importantly, in the left-hand side panel, we knew from the beginning that when mice are sleep deprived and they are sleepy, this T221 phosphorylation levels goes up. Okay, so this seems to be the physiological regulation representing the level of sleep need. Unfortunately, yeah, it's very weak. But anyway, okay, so we try to examine the importance of this T221 phosphorylation. In order to do that, we induced T221A point mutation on top of sleepy mutation, and this T221A mutation negates the effect of original sleepy mutation, okay? Showing that this T221 phosphorylation or the kinase activity of this enzyme is important. Also, then we knocked out LKB1, the potential upstream kinase, by the same inducible system, and as expected, we see a great reduction of sleep need index as well as the increase in wake time or decrease in non-REM sleep time. And on top of that, when we knock out LKB1, then the SIK3 sleepy phenotype is blunted. On the other hand, when we induce T221E mutation, which makes it partially constitutively active kinase, that also blunt the effect of LKB1 mutation, genetically proving that SIK3 is certainly target for LKB1 in terms of sleep regulation. How about downstream? So we continued this forward genetic screen, and we found this sleepy2 pedigree, which has very similar phenotype to original sleepy mutants. However, it does have a totally different genetic loci, and it turned out that sleepy2 mutants carry loss of function mutation in what's called HDAC4, histone deacetylase number 4. So this is near knockout hypomorph of HDAC4, heterozygous. As you may know, these HDAC4579, these proteins does not really work as a histone deacetylase. Rather, its name is misnomer. Rather, they work as a transcription co-repressor in the nucleus. And importantly, literature suggests that this S245 residue, this serine residue, is actually target for SIK. So we are very excited. This might be simply the direct target for SIK3. Indeed, the either heterozygous or homozygous knockout of HDAC4 or 5 increase sleep time and sleep need. Double hetero is kind of works additively. So it suggests that total amount of HDAC proteins might be important. And also pharmacological inhibition of these proteins also increase sleep. So that's fine. It's well known actually that phosphorylation of this residue kicks them out of nucleus. So they can't no longer work as a transcriptional repressor. So maybe the nuclear amount of HDAC might be important. So we actually measured nuclear amount levels of HDAC4 proteins in various conditions, and lo and behold, whenever mice are sleepier after sleep deprivation or in the daytime or rather than nighttime or under sleepy mutation, always the nuclear level of HDAC4 is decreased endogenously. So indicating that indeed nuclear level of HDAC4 represents the level of sleep need. And importantly, as I said, HDAC4 is a transcriptional repressor, so using, this is a HEC cell, HEC293 cells, we are measuring the MEF2-driven level of transcription by luciferase reporter gene. And the MEF2 transcription at the baseline is this, and then HDAC4 strongly suppress that transcription as known as in the literature. And important for us, the wild-type SIK3 derepress this action by phosphorylating that S245 residue. And importantly, sleepy type of mutant SIK3 further derepress, and kinase dead version of SIK3 does not work. So we mutated this S245 residue to alanine, and this derepression goes away. And importantly, the level of this phosphorylation is indeed increased in sleepy mutant in vivo brain tissue, like you see here. So finally, we then made HDAC4 S245A mutant mice, and lo and behold, they do sleep less. This is awake time, sleep less, and the sleep need index is also greatly reduced. And also, this S245A mutation in HDAC4 gene suppresses the effect of sleepy mutation. Again, suggesting this genetic epistasis relationships. Proving that indeed HDAC4 S245 is target for the SIK3 kinase activity. So this is a semi-conclusion. So SIK3 pathway, this LKB1 SIK3 HDAC45 pathway represents the level of sleep need in vivo and regulate the sleep accordingly. So original sleepy mutation is a gain of function mutation which kicks out HDAC45 from the nucleus, which promotes sleep. And sleepy2 mutation is a heterozygous loss of function mutation in HDAC4, which mimics this original sleepy mutation, and the upstream kinases include LKB1 and PKA pathway here. This, function of PKA pathway is still very much unclear at this time. So what are these downstream target transcripts? So we did some initial single-cell RNA-seq experiments showing that certain, subsets of transcripts are upregulated in the cortical glutamatergic cells when they are sleepy, and as well as they are exposed to sleepy mutant version of SIK3. Such common transcripts, increased transcripts, include some interesting things such as BDNF, which is known to be indicator of a level of sleep need already. And finally, as I already said, this same molecular pathway also regulates the timing of sleep through the regulation of suprachiasmatic nucleus as well. So, oops. Now it's stuck. Could you help? Oh, thank you. Oh, yeah, that's fine. This is not such important thing. The detail is in these two papers published end of last year in Nature. So could you go forward? Yeah. So amazingly, this same pathway, HDAC4, SIK3 pathway, works to regulate sleep even in C. elegans, the worms, nematodes. So in this case, knockout of SIK3 homologue decreased their version of sleep behavior. HDAC4 knockout increased sleep, and double knockout is like downstream knockout. So it's exactly the same as mouse pathway. So these are published in these two papers. And finally, among other things, one of the things we are doing right now is this suppressor screen in yeast or Drosophila genetic style. So we are mutagenizing the sleepy mutants further randomly by ENU and screening the mice which has a normal sleep pattern despite the fact that they are sleepy mutants. So this is a classical suppressor screen, and we indeed found several mutant pedigrees with a apparently normal sleep time despite sleepy mutation. So we are mapping the gene right now. So that's about it for today's presentation. This is a just short acknowledgement list. And finally, when we are measuring, in the last 10 years or so, we measured more than 20,000 mice, sleep-wake of 20,000 mice. And when we are doing this type of thing, we thought that we might be able to do the same thing in humans. So unfortunately, as you may know, sleep measurement, official sleep measurement in humans takes so-called polysomnography, which is very, very cumbersome, usually, many times requiring hospitalization, and you will have to sleep in a spaghetti, kind of a tier. So we recently developed this type of, very easy in-home sleep EEG measurement device, and this device itself is a so-called IoT device, and instantaneously your sleep EEG is converted into sleep states. So that type of thing we are doing as a kind of real-world implementation type of application studies as well these days. Okay, so that's it, and my final slide here acknowledges our institute. So this is a very long name, so we call ourselves Triple IS, IIIS, or Triple IS, and these are PIs, our PIs. So we claim ourselves as the largest institute which is specialized in the basic research of sleep and these days hibernation as well. So this is our building. So if you have a chance to come to Japan, please stop by in Tsukuba to visit us. Thank you for your attention and sorry for this lots of trouble.
So Masashi, we're slightly in difficulty because we've run through, because of the technical difficulties, our question time. I wonder if you're happy to stay here and just personally answer questions from people. No problem. Those who feel they need to leave, feel free. If anyone would like to ask a question, I can see there's maybe at least one or two, so we'll take one or two questions.
Okay, thank you.
Can we have the... is it number 1 or 2?
Hello. Yes.
Yeah.
Okay.
Hello.
Kevin Shang from UC Davis. Beautiful work. And you mentioned earlier orexin mechanism. So have you ever been able to link the orexin regulation to later mutants? It seems like they are in the same pathway.
Okay. Thank you. Okay, so we think orexin is a component of this switching machinery, the neural circuit-based switching machinery. And I don't think orexin by itself directly participate in sleep homeostasis. For example, these orexin-deficient patients or mice do not have a difference in total 24-hour sleep time. The problem is their sleep is very unstable. Sleep-wake is unstable. That's the problem. But total sleep time does not differ. So
the water parallel in your cartoon is beautiful. So I think Dan Yang in Berkeley has a paper showing ATP accumulation in the brain over the daytime. It's one of those triggers. So what do you inside? Can you comment that and maybe a little bit broad spectrum? What other things can be a water?
Right. Okay. Yeah. I mean, probably water could mean multiple things. Yeah. It's such a deep problem. I'm struggling to solve. Okay, thank you.
Thank you.
Okay, I'll take can we have, I know it's a complex subject, but can we have shortish questions and short answers?
Great presentation, Masashi. Is there any evidence that people suffering from hypersomnia have defects in SIG3 or in HDAC4 or 5, or is this unrelated?
Thank you. So far, we do not see any meaningful mutant alleles in these genes. It's just, doesn't happen yet, right. Or nobody has found.
Or the other way is the orexin system implicated in any way if SIG3 is mutated or if HDAC, or for that matter, histamine or adenosine, I don't know.
Right. So far, as I said, none of the idiopathic hypersomnia patients are reported with these mutations yet.
I'm going to take the last question.
Okay.
I'm really sorry, but somebody has to be in here within 5 minutes for the next session.
Okay.
You showed a lady with cataplexy, and it was absolutely beautiful. Clinically, we use low-dose antidepressants to treat cataplexy, and it normally just stops the cataplexy. Have you got any idea how that works?
Yes. Actually, my colleague, Takeshi Sakurai, recently published this very important paper in Science showing that this non-REM to REM transition is regulated by dopaminergic signal in amygdala. Very unexpected place. And when this amygdala sees the dopamine, whenever sees the dopamine surge, the animal going to REM sleep-like state. And that surge, in normal animal, that surge happens only during non-REM sleep, never in wakefulness, okay? That's called REM gating. However, under orexin deficiency, that dopamine surge in amygdala can happen during wakefulness. And that leads to the direct transition from wakefulness to REM sleep, which is cataplexy. And importantly, what orexin does is to suppress that dopaminergic signal probably through serotonergic signal. And that's why those antidepressant works to prevent cataplexy. So that's a kind of downstream of orexin signal. And antidepressant, by boosting serotonergic signal, it prevents cataplexy. That's our current thoughts. Thank you for that important question.
Thank you, Masashi. Thank you to the audience. We'll close this session.
Thank you so much.
Make a start. Apologies for the delay. The previous session went over time. My name is Ross Vlahos. I'm a professor of pharmacology at RMIT University in Melbourne. You'll see that I'm wearing an Tsukuba hat, wearing a blue chambray shirt. So given that I've got the chairman's prerogative, it's just to advertise that we're holding the World Congress of Pharmacology in 2026 in Melbourne, Australia. So hopefully I'll see some of you, if not all of you, there. So without further ado, I'd like to welcome Manon to the stage, who will talk about quantification of systemic short-chain fatty acids and lipopolysaccharides for early detection of intestinal permeability in transplant patients.
Hi everyone, thank you for the committee of the Congress to let me present you my work entitled the quantification of systemic short-chain fatty acid and lipopolysaccharides for early detection of intestinal permeability in transplant patients. First, a little bit of context of solid organ transplantation. So the transplantation is a replacement of a damaged organ, and this improves the quality of life of patients suffering from terminal diseases. Worldwide, the most transplanted organ is the kidney, which is followed by the liver. The transplanted patients need a lifelong immunosuppression protocol to avoid graft rejection. This immunosuppression protocol includes calcineurin inhibitors, such as tacrolimus and cyclosporine. They also include mTOR inhibitors, like everolimus and sirolimus, antiproliferative drugs, like mycophenolic acid and azathioprine, and they may also include monoclonal antibodies and glucocorticoids. However, these transplanted patients may have infectious and tumors, hematological nervous system and vascular disorders, such as hypertension. They also can have kidney disorders like nephrotoxicity. It's the case for calcineurin inhibitors, such as tacrolimus and cyclosporine. And they also can cause metabolism and nutrition disorders like hyperglycemia and diabetes. One of the major concerns are the gastrointestinal disorders like diarrhea and nausea for mycophenolic acid treated patients. So the mycophenolic acid is the most widely used immunosuppressant in kidney, liver, and heart transplant patients, as well as in autoimmune diseases. This molecule is a selective, non-competitive, and reversible inhibitor of the inosine 5'-monophosphate dehydrogenase type 2, EMPDH2. This enzyme is implicated in the de novo nucleotide biosynthesis of DNA in B and T lymphocytes, and this inhibition by the mycophenolic acid leads to the cytostatic effect on these cells. Unfortunately, 30% of transplanted patients undergo gastrointestinal disorders, toxicity. This is characterized by the apoptotic bodies, like as we can see here, as well as with the recruit, and in the more severe cases, there is ulcers. Our hypothesis is that mycophenolic acid transplanted patients develop intestinal permeability. Indeed, in preclinical models, we can see here that the mycophenolic acid treatment induces tight junction protein alterations, such as Z1 and occludin. And the mice model of MMF, we can see here that the treatment with mycophenolic acid leads to endotoxemia. So our objective of the study was to quantify the bacterial products to evaluate the intestinal permeability in transplanted patients. So for that, we quantify lipopolysaccharides, or LPS. LPS are the component of the outer membrane of gram-negative bacteria, and in physiological conditions, they do not penetrate into the systemic circulation. So the translocation of this LPS into the blood compartment is a biomarker of intestinal permeability. We also choose to quantify short-chain fatty acids that are produced by the gut microbiota from the detailled fiber of the gut microbiota. There are three main short-chain fatty acids, acetate, propionate, and butyrate. They are actively transported to the systemic circulation through the intestinal epithelial cells. They have multiple roles in the intestinal homeostasis and immune system modulations. So to assess the intestinal permeability, we collect plasma samples from healthy volunteers and plasma samples from heart transplant adult patients. These patients include in their protocol, in their immunosuppression protocol, mycophenolic acid, and they are 1 year post-transplanted. After, we quantify lipopolysaccharides and short-chain fatty acids, lipopolysaccharides through ELISA dosage, and short-chain fatty acids through mass spectrometry. And after, we perform a hierarchical agglomerative clustering based on the principal component with our software. According to study population characteristics, the age and body mass index are similar between healthy volunteers and transplant patients, as you can see here. There are 2 women and 80 men in the both population, in healthy volunteers and in transplant patients. And for transplant patients, the immunosuppression include mycophenolic acid and tacrolimus or cyclosporine. According to LPS and short-chain fatty acid concentration, we can see that LPS are 3 times higher in mycophenolic acid-treated patients than in healthy volunteers. According to the sum of the short-chain fatty acid concentration, we can see that it is almost 2 times higher than in transplant patients. After, we decided to do clustering based on these concentrations. And we have a positive correlation between LPS and short-chain fatty acid concentrations. And the hierarchical clustering analysis identified 4 groups. The cluster A in red with 20 healthy volunteers and 9 transplant patients. The cluster B in blue with 7 transplant patients, the cluster C in green with 4 transplant patients, and the cluster D with only 1 transplant patient. According to short-chain fatty acid and lipopolysaccharide concentration of these clusters, the cluster A has low LPS and low short-chain fatty acids. The cluster B has low LPS and high short-chain fatty acid concentrations. The cluster C has high LPS and low short-chain fatty acid concentration. And the cluster D with only 1 transplant patient has high LPS and high short-chain fatty acid. To conclude, on the intestinal permeability in transplant patients, we explore the systemic translocation of bacterial products of LPS and metabolite short-chain fatty acid in a cohort of 1 year post-transplant patient treated with mycophenolic acid and healthy volunteers. As we can see in the cluster A, we have low LPS, so no translocation of the LPS into the systemic circulation, but high and low short-chain fatty acid that is in physiological condition. So the cluster A is the healthy grade, is for us the healthy grade of intestinal permeability. The cluster C is, we think that cluster C is an established grade of intestinal permeability due to the increase of systemic LPS concentration into the systemic circulation. But the low short-chain fatty acid concentration in the cluster C can be explained by an intestinal dysbiosis that might be due to the immunosuppressive protocol, due to the mycophenolic acid, calcineurin inhibitors, and antibiotics. So we here demonstrate for the first time that there is an increase in intestinal permeability in patients treated with mycophenolic acid. However, our results also suggest an intermediate grade of intestinal permeability. Indeed, the cluster B has low LPS, so moderate intestinal permeability, but high short-chain fatty acids that are small-sized bacterial metabolites. And these patients might be at early grade of intestinal permeability. These patients are at higher risk to develop systemic inflammation and metabolic disorders such as diabetes. So we need to focus on these patients to avoid comorbidities in the future of transplant patients. And clinical studies are underway to validate these findings in other larger cohorts. Thank you for your attention.
Okay, thank you, Manon, for a wonderful presentation that was exactly on time, so thank you. Now, are there any questions from the floor? We do have a roving microphone.
Thank you. You missed one cluster. What about the high LPS, high SCFA?
For the cluster D, so we have only one patient. So we think that this patient is in intestinal permeability due to his high level of LPS, but with no dysbiosis due to the elevated concentration of short-chain fatty acid in systemic circulation. But we only have one transplant patient. It's a little bit difficult to conclude.
So, Manon, I have a question around the age and the weight of these patients. So I may have missed it, but what was the average age of these patients and average weight?
The average age was around 40 years old, I think, in the healthy volunteers and transplant patients. And the weight, I don't remember, but it's equivalent between both populations.
Sure. Simone has a question.
I'm sorry, I don't understand.
Hello?
Yep.
Okay. So these were heart transplant patients. Do you expect to see the same results with the clustering if it was a different organ transplant?
Yes, we plan to explore these metabolites in other transplant patients, such as in kidney transplant patients, to conclude if there is an intestinal permeability and if short-chain fatty acids are an early biomarker of intestinal dysbiosis, permeability.
And I just have one other question. So your cluster B population, do they have any other comorbidities that I guess the 9 healthy transplants that were in cluster A that I guess you deemed relatively healthy, do they have any other additional comorbidities that could put them in that cluster B?
Transplant patients has a lot of comorbidity. They have diabetes, hypertension, they may have tumors. So this intestinal permeability has been found in this in diabetes. Someone with diabetes has intestinal permeability and intestinal dysbiosis.
So Manon, I have a question. So the short-chain fatty acids are produced by the gut microbiota. So from a therapeutic point of view, are these patients put on high-fiber diet to replenish or aid in this process?
Usually the transplant patients have a specific regimen to follow, and we expect that if we give more high-fiber diet, it will be better for the gut homeostasis.
Yeah, absolutely. Just I know some work looking at replenishing short-chain free or fatty acids. Sorry, for in the context of respiratory diseases, and modifying diet may have a therapeutic impact on people with asthma, for example.
But there is also the short-chain fatty acid supplementation in inflammatory bowel disease works for example, butyrates works for reestablish the intestinal barrier with butyrate supplementation. So why not?
Okay, excellent. Thank you very much. Okay, let's thank Anna. Okay, our next presentation is by Thomas, and he will be telling us about changes of short-chain fatty acid levels in inflammatory bowel diseases, including Crohn's disease and ulcerative colitis, in comparison to non-inflammatory bowel disease in view of the samples collected within the Integrative Human Microbiome Project. Thank you, Thomas.
Thank you. Mr. Chairman, ladies and gentlemen, it's my great pleasure to have an opportunity to present to you some results of our recent investigation. The title of the presentation is very long, however, it can be summarized in underlined 3 sentences, words: fatty acids, inflammatory bowel diseases, and integrative human microbiome project. So, I'm from Gdańsk, from Poland. I'm pharmacist. I'm from Faculty of Pharmacy. I'm the head of Department of Pharmaceutical Chemistry. And as a researcher, in fact, I'm analytical chemist. And I'm analytical chemist who is involved in a couple of projects related to pharmaceutical and biomedical analysis, to analyze drugs and clinical samples, to search some potential biomarkers, also with metabolomics approaches. In our group, we have also some branches related to medicinal chemistry, and it's my main research tasks. Besides, I'm also involved in some organization administrative tasks in our university, and also I'm the teacher of pharmacology not only in Gdańsk but also in another city in Słupsk, teaching not only pharmacists but also nurses, medical rescues, emergency staff, obstetrics, and so on. It's also the reason why I'm here to teach maybe something about more inspirations how to teach people pharmacology. Coming back to my research, of course I have papers. As I said, they are mostly related to analytical chemistry. Mostly they are experimentally data-based papers, of course. And they are realized, those experiments are realized in our labs. And here you see some labs which are empty. And of course they are not empty every day. They were, however, empty during COVID-19 pandemics. It was time when I was frightened very much about the production of experimental data for our experiments. And that time I recollected the idea to use maybe available very commonly experimental data in the formula of open access. In fact, we have a number of open access data in the literature which we could use, and that time I have found one paper which was pretty interesting for me. It was published in Nature. It was called Multi-omics of the Gut Microbial Ecosystem in Inflammatory Bowel Diseases. And this paper had an idea to compare microbiome of the people with Crohn's disease and ulcerative colitis in comparison to non-inflammatory bowel disease people. So in this paper, we can find huge amount of data related to the comparison of anaerobes, obligate and facultative, a number of molecular disruptions data on the level of host serum related to antibodies, a lot of molecular disruptions data for metabolite pools. Among them were also short-chain fatty acids, as I discovered later on, fatty acids. So a lot of data can be found here, as I said, and they are part of so-called Integrative Human Microbiome Project, which was dedicated to the collection of samples, almost 3,000 samples, of stool, biopsy, and blood samples from 132 subjects over 1 year. A couple of data are available not only in the paper, but also they are available through the website, which is shown here, related to inflammatory bowel disease multi-omics database. And within this huge amount of data, one can find also data related to fatty acids. What was the interest for me, because I like to consider fatty acids as building blocks of cell membranes, storage materials in cells, but also as products involved in cell signaling. So reading the article, we can find some information on fatty acids. Usually they can be summarized that there are differences between fatty acids in patients with inflammatory bowel diseases in comparison to non-inflammatory bowel diseases patients. However, not very details on individual fatty acids can be found there. Some of them can be found here, as for example, the increased level of arachidonate as a precursor of prostaglandins, as we know, found in people with inflammatory bowel diseases. However, no other detailed data can be found, but in contrary, when we search the database, we can find the number of fatty acids identified in the project, namely listed here 30 compounds. 30 compounds found in more than 500 stool samples identified with LC-MS systems, with sophisticated analytical approaches. Having such data, which I showed here, with chromatographic retention time listed in one of the columns here, of course, my first step was to adapt known for me approach called quantitative structure retention relationships to find the possible relationships between chemical structures for all of those 30 fatty acids, in contrary to retention time, chromatographic retention time. Very nice regression model was derived, but of course this is something what could be extended a little bit more in another step. What is more interesting for us, I suppose, is also the availability of a lot of data for those more than 500 stool samples related to the signals measured for each patient and each control men and women possessing Crohn's disease or ulcerative colitis or non-inflammatory bowel diseases. So the matrix, the data matrix of 29 means fatty acids. I excluded one fatty acid because after QSRR analysis it appeared that one of compound oleanate, namely, was repeated in this table times 546 samples. It's huge amount of data which could be processed with classical statistics. And in our results, with non-parametric tests with Kruskal values, analysis of variance, we have found several statistically significant differences between non-inflammatory bowel diseases and inflammatory bowel diseases patients. Especially for 8 compounds, fatty acids, I focused further on details. And besides more medical considerations, also chemical considerations were done with QSRR, and those 8 fatty acids as the most statistically significant were treated in one path of my research as test analytes, in contrary to other fatty acids considered as model analytes. And with the use of model analytes, we were able to derive the model, regression model, to predict retention time of 8 test fatty acids. As it was done, it was very nice predictions of retention time with perfect order of them in chromatographic runs too. However, coming back to those data which, according to me, are even more interesting, the set of comparisons between Crohn's disease and ulcerative colitis for men and women in comparison to non-inflammatory bowel diseases, men and women, can be found here. And exceptionally, only for caproate, we have found that concentrations were statistically significant for men, for diseased and non-diseased people. However, in such a way that for non-inflammatory bowel diseases, concentrations were even higher. It was something different than was found for the rest, 7 fatty acids considered as the most interesting, like here for arachidonate, when we saw statistically significant differences indicated in red for non-inflammatory bowel diseases, in contrary to both Crohn's disease and ulcerative colitis. In this case, also the differences for Crohn's disease between men and women was noticed. And considering further, another fatty acid was the same statistically significant data, and so on, and so on. The worst data were noticed for eicosapentaenoate, when only for men statistically significant data were noticed in the considering non-inflammatory bowel diseases versus Crohn's disease or ulcerative colitis. So it's next compound and the next, and it's almost all. Adrenate was the last compound fatty acid what we analyzed, and concluding, I have three short conclusions with 3 acronyms OA, QSRR, and FA. In my opinion, open data are huge, still huge, of course, huge amount of information which could be proceeded by everyone, not only to produce new experimental data. Second conclusion is that QSRR approach could be not only useful for predictions of retention times for analytical chemistry, but also in view of the strategy to use it as additional filter constraint to identify compound of interest. And the third, fatty acids as potential targets, as potential prognostics during development of Crohn's disease and ulcerative colitis should be, according to me, taken into account because of distinctively statistically significant changes which could be observed after those data which was processed. Thank you very much for your kind attention.
Thank you, Thomas. Are there any questions? No, Simone.
I guess I can start. So out of the 8 fatty acids that you found were significant, which one do you think would be the first that you would try to use for a target or for a therapeutic target?
Which one could be used for therapy targets? In fact, I couldn't imagine very easily how to answer for this question. Maybe those ones which are more commonly known for people engaged in targeting searching, like arachidonate or adrenate, they, according to me, are more easy to search for another more complex networking with other compounds. So it's what I can say now. Thank you.
Thomas, I may have missed it. You had a significant number of stool samples, and you showed data for male, female. What percentage were male and what were female? Do you know the numbers for those groups?
Frankly speaking, I do not remember what was. Is it proportional number or not? For sure, I should check it just in the paper published in Nature. Statistically, I could expect it's similar proportion.
Yeah. Good. Okay, in the interest of time, we should move on. Thank you very much, Thomas.
Thank you very much.
So I have a question. Is John Gull here? The speaker didn't present themselves to me at the beginning of the session, so I suspect he or she is not here. Which means we can invite Thomas back for more questions. I'm only joking. All right. Okay, if that person's not there, we should go on to the next speaker, which is Yu Jin, and the title of her presentation is Protective Role of Ginsenoside, RH1 on LPS-induced endothelial cell injury through suppressing TLR2/4-mediated STAT3, NF-kappa B, and ER stress signaling pathway. Thank you.
So, okay, thank you for introducing me. So, good afternoon, everyone. My name is Yu Jin from Chungnam National University in South Korea. So, before I start my presentation, I would like to thank to WCP committee to give me the opportunity to present my study. So, today I will present about my research. The title is Protective Role of Ginsenoside RH1 on LPS-induced endothelial cell injury. So actually, the inflammation is a part of the protection, the body protection agent from the allergic or irritant or infection. However, the pro-inflammatory sustained or excessive the excessive and sustained production of the pro-inflammatory cytokines can lead to chronic inflammation, and its activation was induced cardiovascular disease, autoimmune disease, and rheumatoid and cancers. So today I will focus on the cardiovascular disease under chronic inflammation. So actually, the cardiovascular disease is one of the leading causes of the death in worldwide, and especially the progression of the atherosclerosis, the main reason of the progression of the cardiovascular disease, and is caused to the diabetes, hypertension, dyslipidemia with inflammation. So actually, the cellular homeostasis is endothelial cell is very important on the atherosclerosis progression. So because in the endothelial cell is the first cell to directly meet the blood flow, and then when we have some of the toxic agents, or something oxidized LDL or high dietary lipid or hyperglycemia, they have maybe endothelial cells got the stress, and after that, endothelial cells show the dissolution of the cellular homeostasis, such as the ER stress, oxidative stress, and inflammatory burdens. So today, I would like to introduce about the ginsenoside. The ginsenoside is one of the compounds from the ginseng after they got the heating and hydrolysis. And the numerous studies, they are revealed that ginsenoside has the various pharmacological effect on the cardiovascular disease by increasing the antioxidant signaling pathway. So in this research, we are using the ginsenoside RH1. The ginsenoside RH1 is one of the minor ginsenoside, which has the big advantage because they can easily absorb in our human body. The RH1 is the metabolite from the RG2 and RE. Previously, our group revealed that ginsenoside RH1 has anti-cancer effect, anti-asthmatic effect, with suppressing angiotensin II-induced vascular smooth muscle cell migration and proliferation. So therefore, we investigate what kind of the signal pathway involved in LPS-induced EC inflammation, and investigate the effects of the RH1 on the LPS-induced EC injury. So before we starting checking the effect of the RH1 on the LPS-induced signal pathway, we checking what kind of the signal pathway involving. So as shown in figure A, as shown in here, the activation of the STAT3 phosphorylation was peaked at the 5 nanogram per ml of the LPS, and the other things, most important inflammatory mediators such as the NF-kappa B and its target molecule, the BKM1 and IKM1 are the increased by LPS treatment time and dose dependently. And next, before we checking the mechanism study, we are using the LPS conjugated with the green fluorescence for checking their epitope of TLR2 and TLR4. As shown in figure A and B, after we treat the LPS conjugate with green fluorescence, the TLR2 and TLR4 fluorescence intensities are increased. And however, before we treat the LPS, RH1 significantly blocked the green fluorescence, and intensity of the TLR2 and TLR4s are significantly suppressed. Mechanistically, as I shown to you, the STAT3 phosphorylation caused by the LPS, they are shown ginsenoside RH1 significantly suppressed its activation, and as well as the NF-kappa phosphorylation and its target molecule BKM1, and the other endothelial cell protection agent, phosphorylation of enos, are significantly reversed by ginsenoside RH1. The consistent with the figure 8, the phosphorylation of the STAT3 fluorescent intensity caused by the LPS significantly suppressed by the ginsenoside RH1. The next is the NF-kappa B promoter activity is the very important role on the releasement of pro-inflammatory cytokines. So in figure A, when we check the TNF-alpha luciferase NF-kappa B promoter activation, so ginsenoside RH1 significantly suppressed its activation. And oppositely, so LPS suppressed inose promoter activation, however, the RH1 significantly recovered its activation. So next, we are focusing on the what is the relationship between the NF-kappa B and enos. We are using the specific inhibitor for the enos, L-name, and NF-kappa B inhibitor, BAY. As shown in figure C, even we treat the enos suppression, the NF-kappa B signal promoter activations are increased. However, even we treat the NF-kappa B suppression, enos promoter activation was not activated. And then finally, ginsenoside RH1 suppressed an LPS-mediated pro-inflammatory cytokine, including TNF-alpha, IL-1 beta, MCP1, and suppressed adhesion molecules, the BK1, IK1, and E-selectin. And the ginsenoside RH1 the suppressed LPS-mediated cell apoptosis through suppressing the activation of the glycolytic caspase 3 with the cell cycle. So next, I would like to introduce about what is the ER stress and what kind of the pathways are important in apoptosis. After the endoplasmic reticulum got the stress, the three kinds of the receptors are activated, and finally the CHOP are increased, and they are induced the secretion of the cytochrome C and its mediated apoptosis signal pathway. So when we checking how the ER stress can activate it by the LPS, the PERK and CHOP signal pathways are activated, and its activation was suppressed by the RH1. The consistent with the protein expression, fluorescence intensity of the CHOP was caused by the LPS suppressed by the RH1. Finally, I will introduce about what is the MPAS assay. Depends on the blood flow, we can show the different atherogenic area. The one thing is the disturbed floor, and they show the pro-inflammatory, pro-proliferative, and through activating the NF-kappa B and the YAP signal pathway. On the other hand, in the static floor, they show the anti-atherogenic area by suppressing the NF-kappa B or TNF-alpha. So now we are choosing the two different kind of the disturbed floor. One thing is the carotid artery, and the other thing is the lesser curvature area. As shown in control level, we stain the GO1 for indicating the endothelial cells. After we inject the LPS, we can show the distal connection and irregular phenotype of the GO1 with the CHOP and BK1 expression. So however, RH1 significantly blocked the LPS-mediated disconnection of the endothelium and suppressing the CHOP and inflammation. Taken together, ginsenoside can suppress the LPS-mediated inflammation by suppressing NF-kappa B caused to the enos suppression, and then ginsenoside RH1 suppress the ER stress. It causes to the cell cycle arrest and apoptosis. So I'd like to thanks to my Professor Kyung-Sung Ho and my lab member. Thanks for your attention.
Okay, thank you, Eugene, for a fabulous presentation. A lot of beautiful data in there, so congratulations. Are there any questions from the floor, apart from Simone? You're making it difficult for me. No? All right, I have a question. We've assessed the ginsengs and also the ginsenosides in preclinical models of COPD, and we've found that they may have some antioxidant properties. So I'm just wondering, in your case, have you measured any oxidative stress or reactive oxygen species? And if so, was RH1 doing anything to the potentially LPS-induced oxidative stress?
So, yes, thank you for your question. So actually, before we starting the disease research, we want to checking how the LPS and the RH1 can, how LPS can induce the oxidative stress in endothelial cells. So actually, endothelial cells got the stress from the LPS by and releasing the ROS. And then we did not add the data into here, but we also showed, confirm the RH1 suppressed oxidative stress in endothelial cells.
Excellent. Did you measure the levels of nitric oxide in your system?
So not yet, because actually when we starting the RH1 study in different kind of the cells, for example, cancer cells, in case of the ginsenocyte RH1 suppress the tumor cancer proliferation by inducing the mitochondrial ROS production with the mitochondrial dysfunction also.
Excellent. All right, any other questions? I guess, Simone, yes, please.
Hi. I was just wondering, all of the work was shown on bacterial mimetic LPS. Have you looked at assessing any viral mimetics, such as like poly-IC, and whether there would be similar or dissimilar outcomes?
I'm sorry, I cannot.
So did you look at, or have you assessed any data using poly-IC, a viral mimetic?
No, I don't think so.
Okay, I think we're out of questions. Okay, please join me in thanking Eugene Chin for a wonderful presentation. Thank you. Now, I do have something. Jian Gao is not here because it's going to be a video presentation. So please stay, turn off the lights, enjoy the movie. And unfortunately, we can't ask questions, but let's please play the video.
Thanks for the introduction of the host. Good afternoon, ladies and gentlemen. My name is Jian Gao. I came from Shanghai Children's Medical Center, School of Medicine, Shanghai Jiaotong University. Unfortunately, due to the visa delays, I am unable to attend the Glasgow conference video offline. My lab is interested in exploring novel molecular mechanisms of pulmonary fibrosis, aiming at developing potential anti-fibrosis bioactive components. It's an honor to have the opportunity to share our work at WCP 2023. My report today is titled NR2 Antioxidant Pathway Suppresses Lung-Mediated EMT. My report includes the following sections. The first part is background. Pulmonary fibrosis is a chronic interstitial tissue lung disease, typically characterized by excessive production and deposition of ECM and remodeling of abnormal lung tissue structure. The pathogenesis of PF is not well understood, and there is no cure for pulmonary fibrosis. Therefore, new therapeutic strategies for its prevention and treatment are urgently needed. EMT serves as one of the most important avenues for production of mesenchymal cells and excessive secretions of ECM, which promotes the development of PF. However, there is still lack of comprehensive understanding of molecular mechanisms of EMT during the development of PF. Many studies have confirmed lung protein has a close contact with EMT. LAM knockdown promotes EMT in breast cancer or renal fibrosis. Moreover, LAM suppresses E-cadherin expression to induce EMT in response to TH-beta-1 signaling in renal fibrosis. However, the role of LAM in the development of EMT during PF has not been clarified yet. In addition, oxidative stress is one of the most important inducers in the pathogenesis of PF. The mechanism of OS promoting the PF involves multiple pathways. NR2 is a significant transcription factor for regulating OS. We previously showed that NR2 blocked EMT progression in BLM-induced PF model. Meanwhile, associated study has reported that LAM acts as a protective molecule on OS agent. Puromycin amylonucleoside-induced apoptosis in human renal tubular epithelial cells. However, the link between OS and LAM is rarely reported in PF, and the potential contact between LAM and NF2 antioxidant pathway has not been verified yet. The second part is hypothesis. Based on this evidence, we hypothesize that LAM involved in EMT-mediated PF and NF2 antioxidant pathway makes an inhibitory role on EMT-mediated PF by suppressing the expression of LAM. The third part is results. In vivo PF model, we first observed the abnormal pathologic changes after BLM administration, and these changes were more severe in NF2 clocked out mouse, indicating that NF2 played a protective role in PF. Moreover, the expression of E-cadherin was further reduced in BLM groups with upregulating expressions of LAM and alpha SMA. Meanwhile, NRF2H01 and NQ01 were stressfully upregulated on day 7 in BLM group, and LAM expression was increased in BLM-treated wild-type and NF2 clocked out groups, with a more robust increase observed in NF2 clocked out mice. These results suggest that NF2H01, NQ01, and LAM participated in the progression of BLM-induced PF, and LAM expression is modulated by NF2. Previously, we observed LAM was upregulated in fibrosis model with emergency of EMT. Thus, we speculated whether LAM could promote the process of EMT. The results show that TH-beta-1 induced the expression of LAM in a dose dependent manner. Next, we silenced them by siRNA before TGF-beta1 stimulation. The process of EMT has been alleviated with the increase of E-cadherin and the decrease of alpha SMA. The above results suggested LAM may be a positive participator protein involving in EMT-induced PF. In vitro experiment, NF2 activator SF8 pre-treated cells before TGF-beta1 stimulation. In both cell lines, E-cadherin was reduced and alpha SMA was increased in TGF-beta1 stimulation group with down-regulated levels of NRF2-HO1 and NQ01, but pre-treatment with SF8 alleviated TGF-beta1 induced EMT. Additionally, SF8 promoted the expressions of NRF2, HO1, and NQ01 and reduced the expression of LAM. Moreover, when RLE6TA and A549 cell lines were transfected with NRF2 siRNA before TGF-beta1 treatment, the expressions of HO1 and NQ01 were reduced, and we observed TGF-beta1 induced EMT was aggravated, accompanied with augment on the expression of LAM. At the same time, the expressions of LAM respectively upregulated and downregulated by TGF-beta1 and SF8 stimulating and further increased when NRF2 was silenced before TGF-beta1 treating. Moreover, we magically found that LAM expressed in nucleus and cytoplasm after TGF-beta1 stimulating, but pretreatment with SF8 would lead to the most of the LAM expression in cytomembrane. This data demonstrates that activating NRF2 antioxidant pathway attenuates TGF-beta1 induced EMT and the expressions of LAM. Next, we further investigate whether NRF2 antioxidant pathway block the process of EMT via LAM. Surprisingly, the protective effect of SF8 against EMT in silenced LAM group was mitigated compared with unsilenced LAM group, which was reflected on the upregulation of alpha SMA and the downregulation of E-cadherin. This suggests that LAM took participate in the process of NRF2 antioxidant pathway, ameliorating the progression of EMT. The first part is discussion and conclusion. In summary, in this study, we provide the advanced evidence that LAM involves in EMT-mediated PF and NRF2 antioxidant pathway makes an inhibitory role on anti-mediated PF by suppressing the expression of LAM. NF2 pathway and LAM may represent potential therapeutic targets for PF treatment, which lead more study to further investigate. We thank for Professor Peng Cao and Chuanping Hu for presenting NRF2 knockdown mice for our experiment. This work was supported by National Natural Science Foundation of China. Welcome to submit your research paper to APSB. And welcome to Shanghai, China. And thank you.
Okay. Thank you, Professor Go, and it was a shame he couldn't be here in person. Obviously, there'll be no questions. So, look, I'd like to thank you for attending this session. I'm sure you will all agree that it's been an informative and exciting session. I'd like to thank the audience for your participation, and in particular, Dr. DeLuca for the numerous questions. And finally, if you can all join me in thanking and congratulating today's speakers.